Hello, and welcome to the second in a series of 4 RGA webcasts examining advances in statistical genetics and the potential that might arise from those advances for anti-selection in the protection industry. In this second webcast, we're joined by Dr. Richard Russell, lead health data scientist at RGA, who will talk to us about the basic genetic framework, how that has evolved into polygenic risk scores, and talk about some of the applications of polygenic risk scores in medicine.
Right, so the human genome is about 3 billion letters long, and we call those letters base pairs. As humans, we have 23 pairs of chromosomes, which is how the DNA is packaged. We inherit 1 chromosome from our father and 1 chromosome from our mother. Now, by far the most common sort of variation that we see in individuals as humans is known as a single nucleotide polymorphism, or a SNP. And those SNPs are actually incredibly common. So as humans, we share 99.99% of our DNA sequence, but it's just a fraction, that 0.01%, that characterises everything about us, our risk to disease, what we look like. And that variation usually comes about as a SNP. Scientists have been studying single nucleotide polymorphisms, or SNPs, for over a decade, and the way that the geneticists have been doing this is using something called a GWAS, or a genome-wide association study. And what that simply does is takes the DNA sequence of people that exhibit a certain trait or have a certain disease, and it compares that against people that don't have that disease or don't exhibit that trait. And we can do that using something called a DNA chip or a DNA microarray. And that might be millions of SNPs long, and as Peter mentioned in an earlier webcast, it's very similar to the DNA chip that 23andMe use, which has actually 600,000 rows of data, or 600,000 SNPs. Now, when we're comparing millions of SNPs and looking for significant ones associated with the disease or trait, we usually use a very low p-value, something that exhibits very high significance. And that's what we're showing on the right-hand side of the plot. We're looking at millions of SNPs across all chromosomes in the humans, and we're saying which ones are highly significant, which ones do we think with high confidence are associated with disease. The first GWAS was conducted in 2005 and looked at an eye disease, AMD, age-related macular degeneration, and that looked at just almost 100 people with that eye disease and 50 people without the eye disease. And that study identified 2 SNPs with very high significance that seemed to be associated with having the disease. Now, since that study conducted over a decade ago, scientists have conducted tens of thousands of GWASes, and each one has identified various variants, various SNPs associated with disease, and they're all catalogued for us to be able to understand exactly how that variant affects disease and what's the biological cause for it. Now, since the first study conducted in AMD, GWASes have grown massively in size. So instead of looking at 100 people with a disease, we're now looking at anywhere upwards of 1 million people, sometimes a couple of million people. And by doing that, we're growing our sample size, we're able to find even rarer genetic variants associated with disease. One of the important things that Peter has mentioned in earlier webcasts is the fact that we're moving on in terms of our understanding of statistical genetics to not just look at genetic variants that are very rare, that have high penetrance. We're starting to look at those variants that are much more common, those that are identified by GWAS studies, and then understand how they, which albeit low penetrance, they have a small effect, a deleterious effect, or a small positive impact on your predisposition to disease or certain traits, but they are far more common. If we think about that common variant and contrast that with less common variants, breast cancer is a very good example. So I think we've probably all heard of the BRCA1 or BRCA2 genetic variations. So they are mutations in the BRCA gene that confer a very high risk of developing breast cancer in your lifetime. So we call that risk a high penetrant genetic mutation. So what does that mean? It means that if you carry that mutation, the lifetime risk of developing breast cancer is much greater. But actually, the thing about these genetic mutations are that they're incredibly rare. So BRCA1 and BRCA2 occurs in about 1 in 500 people in the UK, What we actually see when we do these GWAS studies is hundreds, thousands, sometimes millions of genetic variants that appear to be associated with disease, but they have very low penetrance. So again, what we see is they have a small increased risk or decreased risk of having a disease. And we call this the common disease, common variant model, because actually most common diseases aren't characterized by high penetrant rare genetic mutations. They're instead characterized by a polygenic architecture. That is, a number of variants appear to add up to give us an overall risk of having a disease. As our understanding of this common disease, common variant model improves and we identify more and more genetic variants, more and more SNPs associated with disease, we're really starting to understand how that polygenic model comes together. And indeed, in the last few years, statistical geneticists have come up with something called a polygenic risk score. And that's simply a way of adding all these common variants together to create an overall risk score to identify those people with a higher risk of disease or indeed a lower risk. Returning to this idea of GWAS studies, what they again allow us to do is identify those SNPs, those common variants associated with disease. And I said earlier that often we choose a very low p-value, we need a very high significance to identify the variants that may be associated with disease. Well, actually, polygenic risk scores do this slightly differently sometimes. We can relax that significance value, we can increase the p-value, and by doing so, we're obviously identifying more and more variants that are associated with a disease or a particular trait. And what the polygenic risk score does is it simply adds all of those together. So if there is a small increased risk of disease or a small decreased risk of disease, we times that effect size by the number of variants that you carry, we add them all up and we get a polygenic risk score, which generally follows a normal distribution. Although the polygenic risk scores are relatively new to statistical genetics, the literature demonstrating how useful they are to predict disease is vast. So here we're looking at just a sample of papers that have used the polygenic risk score to differentiate disease risk. And it's lots of common disease. So it's coronary artery disease, it's type 2 diabetes, it's stroke, and it's specific cancers. And actually these polygenic risk scores are demonstrating huge, powerful risk differentiation. So typically what we can do is we can compare people that have the highest polygenic risk scores, the highest genetic predisposition to a disease, and compare it against people who have the lowest polygenic risk scores, have a lower genetic predisposition to disease. And what you'll see here is that we're seeing very powerful risk differentiation between those two groups. And you'll also see that the number of genetic variants being used in polygenic risk scores varies from tens of SNPs to hundreds of SNPs to thousands and sometimes actually millions of SNPs. So it's not just common disorders that we're looking at, we're also looking at less common disease, we're looking at particular traits. There was a polygenic risk score developed for insomnia recently using a big GWAS study of a couple of million of people. And again, they're being published in very important papers in very high impact journals. So the literature is moving from stats genetics to clinical medicine. And obviously, if we do demonstrate that polygenic risk scores are powerful risk differentiators, then that's important for the clinicians. Although polygenic risk scores have only been around for a few years, they're already demonstrating very powerful risk differentiation. So in this study by Abraham and colleagues from 2015, they developed a polygenic risk score for coronary disease and then investigated in independent data sets whether it was a powerful risk differentiator. So what they did was they looked at various cohorts of men and women, notably the FinRisk cohort and the Framingham Heart study, and they used that polygenic risk score alongside the best clinical risk information for coronary disease, i.e. your BMI, your blood pressure, and most notably family history. And after controlling for those risk factors, the polygenic risk score still proved to be an extremely good risk differentiator of developing coronary disease. If we look at one of the particular cohorts, so the Framingham Heart study, and males specifically in that heart study, what we can do is plot the trajectories of people that go on to develop coronary disease according to their age. And as I said earlier, typically what we do is we look at those people with the highest risk scores and we compare it against people with the lowest risk scores. So here, the red line is men with the highest polygenic risk scores, the top 20% of polygenic risk scores. And we're plotting how long it takes until 10% of those men have had a coronary event. And what we see is that risk level is achieved at 51 years of age. So men in the Framingham Heart study with the highest polygenic risk scores, 10% of them have had a coronary event by the age of 51. If we contrast that with men with the lowest polygenic risk scores, i.e. their lowest genetic predisposition, we see that 10% of those men don't achieve that risk level until 63 years old. So there's a very significant difference in the risk trajectory there, 12 years in fact. In this same paper, what the scientists did was look at a different cohort of men in the FIN-RISK study, and actually for that same differentiator, we see an 18-year gap. An important question is, how does your polygenic risk score interact with your lifestyle risk factors and other risk factors? So this was a really important paper published in 2016 in the New England Journal of Medicine by Kira and colleagues. And what they did was look at a polygenic risk score for coronary disease, but also look at the lifestyle risk factors that you have. And they categorized people as living a favorable lifestyle, an intermediate lifestyle, or an unfavorable lifestyle according to their BMI, their diet, their exercise, and their smoking status. And it was a really interesting study because there was a powerful message there, which is your genetics is not deterministic. So even though you might have a high genetic predisposition to coronary disease, if you live a favourable lifestyle, you can lower your absolute risk of having a coronary event. And the other interesting conclusion from the study was it was largely independent in terms of your genetic risk and your lifestyle risk. These polygenic risk scores hold lots of promise for clinical medicine, and an obvious way of utilising the risk information is in cancer screening. There is a polygenic risk score that's been developed for your risk of breast cancer, and we've talked about that disease and the genetic predisposition to it in some detail. But obviously, if you have a high predisposition to breast cancer coming through from your polygenic risk score, then perhaps what we should be doing is screening women with that high genetic predisposition sooner. In the UK, the typical age that a woman is screened from is 47 years old, and that's just based on an average risk level amongst UK women. But if we were to take those in the highest risk percentiles according to their polygenic risk score, we'd actually see that they achieve the same risk level much sooner. In fact, in the top 5% of polygenic risk scores, women reach that same risk level at 37 years old. So really, what we could do is say, actually, if you have that high genetic predisposition, we should see you sooner for screening. The risk of breast cancer is very much dependent on a number of genetic mutations. That includes the BRCA1 and BRCA2 mutation, and obviously now, as we've been discussing, the polygenic risk score, the polygenic architecture that can underpin breast cancer. In the UK, about 1 in 8 women will have breast cancer in their lifetime, so the penetrance there, as we discussed earlier, is about 12.5%. If we look at women who have the BRCA1 or BRCA2 mutation, then the penetrance is much higher, somewhere around 80%. So a much higher risk of developing breast cancer if you carry that BRCA1 or BRCA2 mutation. The prevalence of the BRCA1 and BRCA2 mutation in the UK is about 1 in 500, so 0.2% of women carry that mutations. And a lot of those women unfortunately do go on to develop breast cancer. But if we look at women who have developed breast cancer, actually only somewhere between about 5 and 10% of them carry one of these high penetrant or moderate penetrant genetic mutations that confer a higher risk. And BRCA1 and BRCA2 are just a couple of the genetic mutations that are deemed high penetrant in breast cancer. So amongst those women who have developed breast cancer, only 10% of them, let's say, carry one of these moderate or high penetrant genetic mutations. So in the other 90%, that may have a very significant family history of breast cancer. What's underpinning the genetic components there if it isn't a moderate or high penetrant genetic mutation? One of the reasons that women with a family history of breast cancer may develop breast cancer is due to the polygenic risk scores, or the polygenic architecture of the disease. And indeed, Myriad Genetics, a diagnostic company in the US, have tapped into this knowledge actually. So what they offer is a genetic test for breast cancer or cancer generally, and they look for these high or moderate penetrant genetic mutations using a gene panel, so an oncogene panel. So we look at 20 odd or so genetic mutations that are known to be significantly associated with cancer, and then we test for those genetic mutations in women who are concerned about their risk of developing breast cancer, possibly because they have a strong family history. However, if a woman takes that oncogene panel test and tests negatively for carrying one of these moderate or high penetrant mutations, what Myriad are offering since September last year is a follow-up test which includes a polygenic risk score for breast cancer. So you don't carry one of these moderate or high penetrant mutations, but your polygenic risk score may confer a higher risk of developing disease. And what they're doing is combining the polygenic risk score with the Tyrer-Cuzick model for developing breast cancer. So all the best kind of clinical information, as well as your family history, which in the case of a breast cancer polygenic risk score does affect the risk differentiation coming from that genetic information. The Myriad polygenic risk score is composed of 86 SNPs, so they use 86 genetic variants to develop this risk score. As I say, they combine that with a Tyrer-Cuzick model and your family history, and then they display your overall lifetime risk of developing breast cancer in a format like this. So as I said before, about 1 in 8 women in the UK or North America are at risk of developing breast cancer in their lifetime. But if you carry certain genetic variants, certain SNPs that increase your risk, then actually you may be at a much higher risk of developing breast cancer. So the penetrance there may be something as high as, let's say, 40 or 50%, just based on the polygenic risk information.
Thank you, Richard. That concludes our second webcast. If you have any questions on this webcast or the other webcasts in this series, please email me at the email address shown on your screen now. If you would like to view the other webcasts in the series, they can be accessed from the website address shown below. Thank you.